this segment we are going to discuss about the development of embryo from diploid zygote. Right? So the topic is embryo development. Right? So the first step that we have discussed is the development of formation of endosperms. In that we have discussed the three different types of endospermic tissues. Once the sufficient amount of endosperm is being developed, then the embryo development will start. So this embryo development is divided into two, that is in case of dicotyledonous as well as monocots. So today in this segment we will discuss embryo development in dicot, that is the dicotyledonous plants. So embryo development in dicots. So I am dividing this into two halves. In this segment the theory part will be written, that is explanation part, and in this segment all the diagrams related to the steps of embryo development will be drawn, and this topic is very important for board as well as for NEET exams, and the diagrams also are very important. So we know that zygote is formed as a result of fertilization between male as well as female gamete, right? So zygote is diploid, is a 2N cell formed by fertilization process, right? Now this zygote will undergo subsequent mitotic cell division specifically, right? This zygote will undergo subsequent mitotic division to form embryo, right? Zygote is a diploid cell and this is forming an embryo which is also again a diploid cell. So of course there has to be mitotic cell division only, right? Gametogenesis process, that is the formation of male as well as female gamete, is by meiotic cell division. But development of embryo, that is formation of embryo from zygote, is always mitotic in nature, that is mitotic cell division. So step number 1. Zygote will undergo unequal transverse cell division. So the first step is zygote undergo unequal transverse, like this. So transverse cell division to form two cells, right? So two cells are formed. The cells are basal cell as well as terminal cells. So zygote will undergo unequal transverse cell division to form two cells. Out of these two cells, we have to label the cells based on the location. This is with respect to the two ends which are present within the embryo sac. If you remember, chalazal end as well as micropylar end. So if we draw the normal structure of the diploid zygote, that is anatropous ovule. If you remember the types of ovules, anatropous as well as orthotropous. In the normal anatropous ovule, the upper end is C, that is chalazal end. Okay, this is C end and this is M, that is micropylar end. So first step is unequal distribution, that is unequal cells are formed as a result of first mitotic division of this zygote. So this is 2N zygote, right? It undergoes transverse cell division to form two cells. One cell is a small cell and another cell is a big cell, right? This is again C and this is again N. The cell which is Now we cannot say upper cell or lower cell. No, this is the wrong way. Now we have to say with respect to the ends, chalazal end or micropylar end. The cell which is located towards the chalazal end is called as terminal cell, right? And the cell which is located towards the micropylar end is called as basal cell. So the cell which is located towards the chalazal C end is terminal, and towards the M end the cell is called as basal cell, right? Now this is the first step. Now the second step is basal cell will divide one more transverse division, that is again mitotic. Now all the division will be mitotic in nature, no meiotic division we will observe. So the second step is basal cell divide transversely to form a long structure, and the name of the structure is suspensor, suspensor. So let us draw the suspensor. So this is the terminal cell and this is the basal cell. Now what will happen? This basal cell will divide. So this is the step number 1. This is step number 2. Basal cell. So terminal cell is as it is. We will see what happens to terminal cell. But now we are focusing on basal cell, right? Terminal cell remain as it is. So this is the C end and I am not labeling the terminal cell again. Basal cell undergoes several divisions to form a long structure which is called as suspensor. So now let us see how this division takes place. So say for example, this is the first basal cell. This basal cell will divide to form 2. So this is the 2 basal cell. These 2 will divide to form 4. So again they will form 1, 2, 3, 4. Now these 4, 1, 2, 3, 4 will divide again to form 8 cell like it. So let us draw few suspensor cells. So this is the structure which is a long structure which is called as suspensor. So this entire structure is called as suspensor. Chalazal end cell, that is terminal cell, is remaining as it is. Now the cell of suspensor which is located towards the micropylar end is called as ostoreal cell. Now what is this ostoreal cell? This is the chalazal end and the last end is micropylar end. So the last cell of suspensor which is towards the micropylar end is called as ostoreal cell. Now this ostoreal cell is big and this is round also. So this is ostoreal, H-A-U-S-T-O-R-I-A-L, ostoreal cell, right? So second step is basal cell divide transversely to form a suspensor, and the last cell of the suspensor is called as ostoreal cell. Now what is the role of this suspensor? Suspensor, say for example, if this is the embryo sac, and within the embryo sac the embryo will be formed after all these steps that we will see. Suspensor cells pushes the developing embryo towards the endosperm. So suspensor cells pushes the embryo towards endosperm in such a manner that developing embryo must receive nourishment for further development. So this is what the role of suspensor is, right? So up till now we have seen the 2 steps. Now let us see what happens to this terminal cell, right? So the third step is Terminal cell will again undergo cell division, but the division is in all the plane, and this structure that is formed after the division of terminal cell is called as pro-embryo. So this is first, second. This part is for diagram, and now the third step. Third step is terminal cell that is located towards the chalazal end. So terminal cell divide vertically in all the plane. That means in all the directions the terminal cell will divide and form a mass of cell. This mass of cell is called as pro-embryo. This is very important stage of development of embryo in the dicot. Right? So terminal cell divide vertically to form a mass of cell. This mass of cell is called as pro-embryo. Right? Now how the division of terminal cell takes place? Say for example, this is the terminal cell that is located towards the chalazal end, right? This is terminal cell. First mitotic division, two cells will be formed. Second mitotic division will result in the formation of four cells, like this we can say, right? 4 will divide into 8, 16, like that. So this structure is called as pro-embryo, and this pro-embryo comprises of 4 cells. So one name is given to this cell and the stage. The cell is called as pro-embryo or pro-embryonal quadrant. Why pro-embryonal quadrant? Quadrant, that means 4 cells are present. So I am going to draw only this cell instead of the regular chalazal end cell, that is the terminal cell. So let us draw the cells 1, 2, 3, 4, and the entire structure is the structure of embryocyte. Now it is not required, so I am removing. Now this structure which is formed is called as pro-embryo. So let us label pro-embryo. This is called as pro-embryo and pro-embryonal octet. Now pro-embryonal quadrant, one more time it will divide to form group of 8 cells, and that is called as pro-embryonal octet. That is group of 8 cells are produced. Right? So this is the third step, that is the formation of pro-embryonal quadrant, and the fourth step is pro-embryonal octet formation. Right? And how pro-embryonal octet will form? This pro-embryonal quadrant will divide one more time to form again mass of cells. This structure is called as pro-embryonal octet. Why octet? Because this structure is made up of, or the embryo is 8 cell stage. Right? So this is pro-embryonal quadrant and this is pro-embryonal octet. So let us draw the third structure, that is the fourth step. So this is first, second step, third step, and now the fourth step will come, right? So this is the pro-embryonal octet 4, 5, 6, 7, right? And the remaining structure will remain as it is. So let us draw few suspensor cells. These are the suspensor cells, and whenever you draw a suspensor, at least 7 to 8 suspensor you draw in the exam. And the last one is ostotrial cell. So this structure is proembryonal octet, right, which is made up of 8 cells. Now this is the fourth step. Comparatively, when this structure will come on to the suspensor, now the suspensor size will decrease. So size of suspensor also decreases. One cell of the suspensor which is in contact with the proembryo will act as hypophysis. So this cell will act as hypophysis. This is the normal ostotrial cell, and this entire structure is the structure of suspensor, and this is the chalazal end, and this is the micropylar end. Okay, so this proembryonal octet is formed. This octet will again divide to form 16 cell stage, and all this division will take place. So say for example 16 cell stage, right? If it is 16 cell stage, then we will obtain 8 octets. That means one group 8 octant, another group 8 octant. This is proembryonal octant, which is formed by proembryonal quadrant. So this is the quadrant stage, and this is the octant stage. Octant stage will again divide from 16 cells. So this 16 cells, so 16 cell embryo can be divided into two 8 cells, that is 2 octets are obtained. Of course, one is located towards the chalazal end, another is located towards micropylar end. These ends are very important in this topic. So the one which is located towards the chalazal end are called as chalazal octet. So they are called as chalazal octet, or you can also say it as terminal octet, because initially the cell was terminal, right? And the one which is located towards the micropylar end is called as micropylar octet. So it is micropylar octet, or you can also say basal octet, because initially the cell which was located towards the end, that is micropylar end, was basal cell. So chalazal octet or terminal octet, micropylar octet or basal octet. Very simple to remember also, right? Now, fifth step, hypophysis will divide to form 8 cell structure, and that structure is also a mass of structure. So actually this is the proembryo quadrant, and proembryo octet is this combination of third as well as fourth step, right? So 1, 2, 3, 4. Now fifth step. So let us go in this direction, that is the fifth step. Fifth step is hypophysis will divide. And what was hypophysis? The cell which is located towards the first cell of this suspensor located towards proembryonal octet, or you can simply say proembryo, which will give rise to embryo. So hypophysis will divide. Hypophysis will divide to form 8 cells.